So good afternoon, everybody. My name is Benedetto Marelli. I'm an associate professor in the Department of Civil Environmental Engineering, and today I'm the host for the Mei Distinguished Seminar Series in this department. And we're very honored to have with us Professor Oded Shoseyov from the Hebrew University of Jerusalem. Oded is a world expert in plant molecular biology, protein engineering, and nanobiotechnology. His work has had a lot of impact in our community. He has more than 350 manuscripts, more than 100 patents. He's considered a serial entrepreneur, as he started more than 20 companies in this realm. He also has a winery. So
you're all welcome.
I'm sure that's the most interesting company for all of us. And he received several awards for his contribution to society, so I don't want to waste more time, and thank you. Please welcome.
Thank you very much. Thank you. I really feel privileged to be here today, and I really thank you all for the invitation and coming. The title of my talk is The Plant Age Materials for the Future, and I hope that by the end of my presentation I will be able to convince you that we are either on the verge of the plant age or already there. But before I start, I'd like to quote the former Minister of Oil of Saudi Arabia, who said something really smart. He said that the Stone Age did not end for lack of stones. And the oil age is going to end long before we run out of oil. And I think for a good reason, because biomaterials are so much better. But this is not good enough for us to criticize the scientists for not making really good synthetic materials from oil. For a simple reason, we didn't have enough time. 200 years of modern chemistry, or maybe 300 years if we account for the Industrial Revolution, is not enough time to develop really good materials. Well, nature had billions of years to develop some of the most amazing materials that we wish we had in our possession. Yet, our friends, orthopedic surgeons, enjoy to glue and screw synthetic materials in our body. And I don't think that this is such a great idea. Why? Because synthetic materials fail. Many of them fail because they are not strong enough for their performance, just like this plastic fork. But admittedly, we have really strong synthetic materials. However, it's still not a good idea to glue and screw them in our body, simply because in nature, things are done differently. In nature, no one takes my head and screw it into my neck. No one takes my skin and glue it onto my body. In nature, everything is self-assembled. Every living organism, as we know, is made of cells. The cells contain DNA. DNA encodes for nanobio building blocks. Many times they are structural proteins. Other times encodes for enzymes that make other things like fatty acids, polysaccharides. And the nice thing about all these bio building blocks is that they don't need us. They recognize each other and self-assemble into scaffolds on which cells are growing, proliferating, organizing into tissues, that organize into organs, and together the organisms is the organism or life, if you like. So we at the Hebrew University in my lab, 18 years ago, we decided to focus on probably the most important nanobiomaterial for us, for human being, and that's collagen. Now, why do I say collagen? Simply because it is the most abundant material in our body. In fact, on a dry basis, it accounts for about 30% of our dry weight. So we have nothing more than collagen in our body except for water. So I give you a few examples. Bone is about 50% collagen, 50% minerals. My skin is about 70% collagen. Tendons are close to 100% collagen. So I always say that anyone who is in the business of replacement parts for human being would like to have collagen because that's a good building block. But admittedly, even before we started our research 18 years ago, there were already in the clinic, in the market, more than 1,000 different medical implants that make use of collagen. You know, simple things like dermal fillers to reduce wrinkles, augment lips, artificial meniscus, bone grafts, and even leaflets for heart valves, all made of collagen. So I guess one of you should ask me then, why bother? You just said that all these products were already on the market. Well, the reason is the source. The source of all these collagen products up until recently is dead bodies, dead pigs, dead cows, and human cadaver. So that's not a very safe source because they all potentially harbor human pathogens. And in fact, the FDA already wrote a notice to all the companies and scientists, asked them, why don't you look for a better and safer alternative? That was already in 2007. So that's exactly what we did. We cloned all the 5 human genes that are responsible to make type 1 human collagen in our body. So the 2 structural proteins, collagen alpha 1, collagen alpha 2, that makes the heterotrimer, but also the 3 post-translational modifying enzymes, the P4H alpha, P4H beta, and LH3, which are required for the proline hydroxylation, lysine hydroxylation, or glycosylation. All are very important for the full decoration or post-translational processing of the protein to make it fully functional. And all these 5 human genes were introduced into a single tobacco plant. So now the plant has the ability to produce human collagen. The technologies were matured. Today in Israel, we grow about 25,000 square meters of greenhouses all over the country, from the Upper Galilee in the middle of the country, also in the southern part of Israel, where it's very hot. The farmers receive these small tobacco plantlets from the company. They grow them for anywhere between 50 to 60 days, depends on the time of the years. We harvest the leaves. The leaves are transported by cooling trucks to the factory, from which we start to make and extract the human collagen. So if you ever made pesto, you know how to make collagen. Essentially the same thing. Just a big food processor, you crush some buffer, extract the juice, concentrate the protein, then the protein goes, the frozen protein goes to the clean rooms in the Rehovot Science Park, where we do all the finer purification steps. The end result is collagen, which is identical to what we have in our body, except better. Why it's better? First of all, plants do not harbor human pathogens, so it is safer. But in addition, this collagen is not extracted from old used tissue. It's brand new. So the 3-dimensional structure of this collagen is much better than the one that is extracted from the old tissue. And from that collagen, we make different medical products. 2 of them already completed clinical trials, reached the market, and I will share with you one of them. Was indicated for diabetic foot ulcer. So diabetes, I don't think need any introduction, except maybe for the fact that more than 450 million people in the world suffer today from diabetes, which means that 20% out of them will experience at least once in their life a diabetic foot ulcer. If we fail to cure this ulcer, it will end up in amputations. The number of amputations per annum is close to 1 million, and that's terrible. It means that every 30 seconds, someone in the world is either losing a toe or their entire foot. So this is the product, or in fact, this is the ulcer. So the doctor is actually debriding the dead cells, so removing the dead cells from the wound, and then inject the human-derived collagen produced in the plant into the wound bed. And now something really nice happened. The live cells in the periphery of the wound, they recognize the collagen because that's their natural scaffold. So they attach to that collagen via special receptors called integrins. And when these integrins get a signal from the collagen, they start to proliferate. and they proliferate and makes granulation tissue. The granulation tissue rise and grow. Once it reaches the level of the skin, another set of cells called keratinocytes start to grow and close the wound.
Question.
Yes?
Why doesn't the immune system attack it? Is it because
Well,
it's dead underneath? Or is
no, it's actually because this collagen is identical to human. So it's human collagen, and there is absolutely no immune response to that collagen. In fact, even bovine collagen, to 97% of the population do not respond to bovine collagen. Okay. So I'm going to share with you a picture. For some of you it may be hard, so you don't have to watch. But this is a case of a male 64-year-old in Italy with one of the doctors that work with this product. And that person suffered from neuropathic leg ulcer with a history of ischemia. And for 2 years, they were unable to cure it. What you see here is the back of the heel, and actually a hole that goes all the way to the bone. So typically, it will end up with amputation. So as a last resort, after debridement of the cells, they injected the collagen, and 3 months later, he is very happy. Another recent development is we took that human collagen and using a simple metacrylation chemistry, we converted it into a photopolymer. So now we can use a DLP printer to print at high resolution collagen scaffolds. In this project, we actually developed a human breast implant. We already completed preclinical trials in small and large animals. The idea in the future is as follows. The patient that suffers from breast cancer will enter the clinic. The doctor will do a PET scan and then decide what part exactly he would like to remove. And then in the operation room, while the doctor is actually performing the surgery, the printer will print exactly the missing match. Now, another doctor at the same time will conduct a liposuction to either from the thighs or from other place to remove some fat tissue, extract on-site adipose-derived mesenchymal stem cells that are then injected into the scaffold and then brought back to the patient. And in a matter of several weeks, the patient actually will grow its own breast. As I told you, we already succeeded in some preclinical trials. This is in a small animal study. You can see on the right-hand side, this is a breast implant of a mouse. And here on the left, you can see 7 days after the cells start to proliferate, in 22 days, it's fully populated with live cells. And of course, breast is just the beginning. We believe that in the future, we'll be able to print some other interesting organs. So I have to admit it took me some time, but 7 years ago, I told myself, hey, collagen is not the end. We can do some other interesting stuff in plants. So we thought about antibodies. Now, why antibodies? Well, I think after the pandemic, everybody are immunologists, so we know that antibodies are important. In fact, I realized the Moderna is not far from here, so everybody understands that humanized antibodies are important and actually serve as really great biological drugs. One good example is Humira. Humira is anti-TNF-alpha, indicated for wide spectrum of autoimmune inflammatory disease, such as rheumatoid arthritis and some other. Great drug, except one big problem. Very, very expensive. The cost per patient per year is about $35,000 US, close to $100 a day, which means that only 1 out of 1,000 people in the world can afford Humira. So it's too expensive. So we asked ourselves, why is it so expensive? So maybe one of you will come and say, well, maybe there are patents. No, patents lapsed. So maybe there's no competition. There is competition. In fact, today there are 5 companies in the world that make the biosimilars. But yet the price didn't drop by more than 15%. The reason why it's so expensive is because of the method of manufacturing. Humanized antibodies are produced by mammalian cells in very, very expensive bioreactors. A lot of stainless steel controls. The media that you have to feed it is also very expensive. Clean rooms, utilities, everything. So it's expensive. So we asked ourselves, well then, why should we produce it in this factory? After all, it's antibodies, so encodes by 2 genes, heavy chain, light chain. Let's just clone the gene, introduce it into a plant, tobacco plant. So that's exactly what we did. And now, grow it at a cost of lettuce or tomato. So we produced the Humira, and in fact, the plant protein, as you can see here in the TNF-alpha neutralization assay, perform exactly like the commercial Humira produced in so much expensive system. So what have we done? We actually took a very complicated and very expensive factory, just cloned the genetic information and introduced it into the farmer. The goal of the company is to produce Humira at a very low cost. In fact, we've been able to reduce the cost of production by 2 orders of magnitude. Our target is not $100 a day, but 70 cents a day. Now, in 70 cents a day, even a farmer in India that maybe makes $8 a day and suffers from rheumatoid arthritis or Crohn's disease, he can afford it. He doesn't really need the government. Trees. Trees are amazing. I don't have to tell you about sequoia. In Israel, I should, but here it's quite obvious. There are huge trees. They carry hundreds of tons for hundreds of years in very cold weather. In fact, even in sun, UV light. I'm not familiar with any plastic chair that can survive such harsh conditions. Yet if you look at the structure of this tree and ask yourself, what is it really made of? How come that it is so strong? So if you look by high-resolution electron microscope, you find that at the end of the day, it is made of nanofibers, about 100 nanometer in length and about 5 nanometer in width, which are very strong, but surprisingly, they're made of sugar. Well, not exactly the same sugar we drink in our tea. It's actually a homopolymer glucose called cellulose, but it's not regular cellulose. It's highly crystalline cellulose. And this highly crystalline cellulose, on a weight basis, is about 10 times stronger than steel. Yet, it's sugar. So analysts all over the world in the last maybe 12, 14 years believe that this material, cellulose nanocrystal, is going to be one of the most important nanobiomaterial for the entire industry. So not just for transportation or construction, but food, sports, almost everywhere. So here's the problem. Say 8 years ago, you wanted to buy half a ton of nanocellulose because you wanted to build a boat or an airplane. You could Google, you could eBay, you could even Alibaba. You couldn't find it. I mean, you would find thousands of papers coming from all over the world, including my lab. You know, we used to say, hey, that's the best material on earth, like we scientists, to brag. But there was no commercial source. So we at the Hebrew University, the Faculty of Agriculture, together with a group of scientists from Finland, Germany, Italy, and Sweden, formed a consortium, one of the European programs. And the idea was to develop a cost-effective method to produce cellulose nanocrystals at a ton level and develop some applications. But the first thing we were looking for is a source of raw material. Obviously, we didn't want to cut sequoia trees, but there is no reason because cellulose is everywhere. In fact, we identified one good source, and that's the sludge of the recycled paper industry. Now, why do I say that it's a good source? Of course, it has a lot of cellulose, but we have a lot of that. In fact, it covers our continents. Europe alone produces 11 million tons of that sludge annually. Just to give you a feel, this is a mountain about 3 kilometers high sitting on a soccer field. Now, not just Europeans make this mountain, the US, China probably few. For everybody it's an environmental problem. For us it's a great source of raw material. So the technology was matured. A company was established called Melodia, and today in Israel we have a production site that can produce up to 200 tons per annum. There is another one by a competing company in Canada, Celluforce company. So now we have a lot of cellulose nanocrystals. The question is, what can you do with that? Well, one very interesting feature of cellulose nanocrystals, unlike CNF, okay, this is cellulose nanocrystal is CNC. CNF is cellulose nanofibers. The cellulose nanocrystals actually form a chiral nematic liquid crystals. Once the concentration is anywhere between 2.5% and 3% in water. So that's beautiful because once you apply these cellulose nanocrystals on any surface, it can be paper, it can be concrete, it can be wood, whatever, plastic. Once the water evaporates, it self-assembles into a very strong and transparent film. Now this film is so strong that it's actually 2 times, the tensile strength is 2 times stronger than epoxy resin. And remember, there is no covalent bond there. It's only hydrogen bonding and van der Waals interactions. Simply the fact that these nanofibers are so well organized, one next to the other, we get these really beautiful results. Now, you can do a lot of interesting things with that. One of them is structural colors. Because cellulose nanocrystals, if you do it right, can generate really beautiful colors, just like we see in nature. And here I can show you a nice picture of butterflies that were made from cellulose nanocrystals. And this is now displayed in the Israeli Museum, did by these 3 brilliant architects and a scientist. So there are a lot of interesting things you can do with these. We picked one which I believe is quite important, and to develop barriers for the food packaging. Why do we need barriers for food packaging? In the old days, we used to pack our food in tin. A great technology because it prevents oxygen, prevents light, so we prevent photooxidation of the food. Problem is tin is expensive. Tin is heavy. So another technology was developed, the flexible packaging. What is flexible packaging? Typically, it's a low-density polyethylene, laminated with aluminum, sometimes PET on top, different combinations. Beautiful technology. By the way, huge, huge market, $1.2 trillion market. Except one big problem. Once you laminated aluminum on polyethylene, there is practically no way back. And this material ends up in the ocean. We already created a new island, the size of New Jersey. If we continue to not do the same, then well, welcome to the new continent, plastic continent. So it's a big, big environmental problem. What we've done, we developed a coating that now is transparent. It's an excellent oxygen barrier because cellulose nanocrystals are fantastic oxygen barriers, but it is transparent. So we included zinc oxide nanoparticles, as you can see, to block just the UV and leave the visible range open. So we thought this is a great packaging. So the client will be able to see the product. Well, it turns out that the industry doesn't want us to see the product, but that's easy to solve. And in fact, Melodia, the company that was established based on that, developed this packaging on the right-hand side, which is regular paper coated with this barrier. And as you can see, it outperforms metallized plastics in terms of oxygen barrier and WVTR. What else you can do with the cellulose nanocrystals? to enforce other materials. For example, we took cotton yarn, the same that my shirt is made of, and we coated it with just 1% by weight of cellulose nanocrystals. And as you can see from the Instron data, the red one is regular cotton yarn, whereas the blue one is the same one coated with the cellulose nanocrystals, added only 1% to the weight, and you can see there was increase in the modulus, increase in the strain at break, and of course, as expected, the toughness is close to 500% increase compared to the other. So what it means is that we can either make the same strength of shirt with 1/5 of the material, or make a much stronger shirt. Another interesting feature of cellulose nanocrystal was achieved when we mixed it with another interesting polymer, which is polyvinyl alcohol, PVOH. And it turns out that if you mix it in water and then you dry it, it forms a very, very strong film that you can see here at the top. You can see here that it's quite transparent and also flexible. But what is more interesting is that here on the left or in the middle, the A panel, this is a cellulose nanocrystals, cross-section without cross-linking. This is cellulose nanocrystals after cross-linking. And here on the right-hand side, this is just polyvinyl alcohol. And here on the bottom here, it's polyvinyl alcohol CNC at a ratio of 1:5, or 5:1, sorry. And here it's 1:5. So 1/5 of the material is CNC and 5 is PVOH. And you can see there is a clear difference. What we see here on the right panel is that we're actually forming very long needles. It turns out that these needles are actually made of PVA, highly crystalline PVA. So the cellulose nanocrystals serve as a template to actually grow very long and very strong crystalline PVA. The most striking thing is the mechanical properties. And I will not go through all of them, but just mention the toughness. This is crosslink CNC, the toughness, and this here is CNC PVA 1:5. Just to give you a feel, the toughness of this material is 3 to 4 times tougher than Dyneema. Dyneema is very high molecular weight polyethylene, which is used as ropes and also some sport goods. So very, very strong material. But sometimes you would like the material to be hydrophobic. And so we've developed some chemistry, very easy chemistry, to acetylate cellulose nanocrystals, make them super hydrophobic. And we've been able to make aerofoams. From here you can see very light. One interesting, and also we can see it's super hydrophobic. But the beauty of that foam is that it has an absorbent capacity for a wide range of organic solvents. Like, for example, here we can absorb 200 times the weight chloroform for 1 gram of the material, of the CNC foam. So you can imagine that we can remove oil spills from the ocean or lots of other things. Another interesting feature of cellulose nanocrystals is the fact that they are actually amphiphilic. They have both hydrophobic sites, but also hydrophilic sites. So taking even material like carbon nanotubes, they are very difficult to be suspended in water. You can add cellulose nanocrystals, either raise some temperature or a little bit of sonication, and you get really nice dispersion. And when you then cast it as a film and evaporate the water, as you can see, you can get even semi-transparent films that are very, very strong. You can see the toughness here is about 7.8 megajoule per cubic meter. Just to give you a feel, this is about 50% stronger than Achilles tendon, the strongest material in our body. And in addition, it is also conducting. So carbon nanotubes are nice, but we really are looking for doing work with 2D materials such as graphene. And I have to say that graphene disappointed us. But new materials like MXenes turn out to be very, very interesting. First of all, MXenes themselves are water suspendable. So that makes it easier. Daniel, one of the graduate students, was able to do both self-assembly with cellular nanocrystals, drop casting, deep casting, and I'm going to share with you some of the results that were submitted for publication but were not published yet. So you can see on the left-hand side at the top, this is self-assembled CNC cross-section in the SEM. You can see the layers. And when you do EDS mapping, you can really see that the MXene is everywhere. When you do drop casting, it's only from the top. Again, in the middle, panel B, you can see the nice organization of the nanocellulose, but the MXene is only at the top. And when you do deep casting, this is right panel, you can see actually that the MXene stays only on the surface, it doesn't penetrate inside. When we take all this information and we actually ask ourselves, so where, just more confirmation of where is everything. So you can see on the right-hand side in blue is the titanium. We have some in the middle and the left, you have a little bit of aluminum, which is part of what's left because, as you know, MXene is made in the assistance of aluminum. In the middle, you can see the carbon, the cellulose, of course, and then the green, the oxygen, so cellulose again. And in the at the bottom, you can see the fluor, the sodium, and of course some sulfur here, which indicates of sulfone groups on the cellulose nanocrystals. So what else we can do with these materials? We can make flexible, transparent, printed circuits which are made of CNC MXene, deep-coated CNC films in MXene, twisted and relaxed, and also foldable materials. You can see here we've been able to fold it into a diameter of close to 40 micron. Admittedly, on the right-hand side, you can see that some of the coating is actually start to fail, but I think that these are quite impressive data. But in order to do real electronics, we need also other materials or other devices like capacitors, for example. So we found out that if we take cellulose nanocrystals and you just mix them with different proteins, proteins that comes from serum albumin, some that come from chickpea, yeast proteins, pea, whatever, canola proteins, we can really get tunable dielectric permittivity and resistivity and essentially make really good capacitors. The one that is actually winning here is the BSA. So now we can really start to realize bioelectronics, if you like. Another interesting application is for LED lighting. A LED light is very popular today because it's very good in energy. It saves us a lot of energy. The problem is that if we really want to have really nice white light, we actually have two options. One, to make 3 LEDs, the RGB, to get the really nice white spectrum, or use blue lead, and then make a coating which is made of rare earth metals, which are toxic and problematic and expensive. So everybody would like to do it with simple organic materials like food grade colors, except for one big problem. Once you shine them in the presence of oxygen, then they photo bleach very quickly. So it's not reasonable. So what we've done here in this paper, we actually took another interesting biomaterial, which is called mucin. Mucin is a very interesting protein. They have both hydrophobic pockets and hydrophilic pockets. So if you mix them with either hydrophobic red color or hydrophilic green color, they will not bind to each other, but they will go to the different pockets. And by doing that, we actually prevent the problem of fret. We want them to be separate. And then now mix all that with cellular nanocrystals and make a film. The cellular nanocrystal hold them in an environment that is actually very protectant from oxygen. So the material that we got is actually quite flexible, and you can see here, this is a regular, on the right, it's on the left, sorry, it's a regular lead. Next to him is one that is coated, and you can see for yourself, the uncoated has this blue light, while the coated has really nice a white light, and we tortured it for a long time in different temperatures and also humidity, and it's really performed well. Another very interesting feature of cellulose nanocrystals is the fact that they are piezoelectric. In fact, in this study, what we have done, we took anodized alumina, introduced into the wells cellulose nanocrystals, and let them self-assemble as nanorods within the alumina. After that, we dissolved the alumina and then produced these really long, if you like, piezoelectric nanofibers. And when we measure them under a piezoelectric measurement device, looks like a specialized AFM, we've been able to measure the piezoelectric properties. Now, of course, came the question, will it work also on a large scale? So in this study, Shiley, one of the graduate students, actually developed just a regular cellulose CNC film, introduce it into an instron, and start to pull and measure the force, which is in the red, can be read by the instron, and using two electrodes, measure the voltage that is produced. And as you can see very clearly, the response curve for the voltage was actually much higher than the one in the instron, which means that it's a very, very, very delicate strain gauge. In fact, Shiley reported that even if someone would open the door in the room, she could see a jump. So it's a very, very sensitive piezoelectric device. Now, in collaboration with Professor Yossi Paltiel from the Applied Physics departments, and admittedly with my son, you can see him here, who is a PhD in this group, they developed an accelerometer/piezo transistor. Now, the design of that is actually very, very simple. What they did was they applied on silicon wafer a very thin layer of gold, so only 100 nanometer. It's about a 1 millimeter thick. And now the idea is that once you apply force, it acts like a gating. And so it's a piezo transistor, if you like. So Omer also developed because his background is a mechanical engineer, so he likes to build things. So he built a really nice device. that enable him, on one hand, to measure the force under constant G. And you can see here some of the data. It turns out that, of course, you can see a voltage response to the applied acceleration. But more interestingly is when he did fast Fourier transformation on that data, he was able to show that the difference between the frequency peaks corresponds to the rotation frequency. So in a way, it is actually not just accelerometer, but also a RPM monitor. With another a graduate student, Dr. Gur Aminadav, he actually PhD before in computer science. He's now doing his second PhD. We've been able to show some really interesting feature of cellulose nanocrystals combined with quantum dots. So you can see the design of the device here. So we have cellulose nanocrystals, which of course have this chiral nematic structure, and in the red quantum dots. At the bottom, you have one conductor, which is the gold, and on the top, we have the indium tin oxide, which is transparent conductive material. And now when we shine it with laser, we can actually do a very effective charge separation. And the reason for this very effective charge separation is because the material is chiral, so we have this chiral-induced spin selectivity that really increase the charge separation. Now, quantum dots that are found in a electric field, they have what is called the Stark shift effect, or quantum confined Stark effect, where the peak of the spectrum is shifted to the red or to the blue. I don't remember. One of them. Actually, it's to the. I see here that it goes to the blue. No, it's to the red. Sorry. I'm probably the best physicist and the biologist in my department, but I'm not a physicist. Anyway, the beauty of that is that this star shift is quite significant. So it's typically rounded and small spherical quantum dots. When they have a Stark shift, it's maybe 0.1, 0.2 nanometer. Here, due to the charge separation and the chiral effect, you can see that the Stark shift is in several nanometers. So that's significant because it means that we can now monitor charge separation of cellulose nanocrystal films from a distance. So that brings a lot of interesting applications. For example, for standoff structural health monitoring. So imagine we just apply it on a bridge or on a train or airplane in other places, and we simply shine with a laser and get real-time information on the strain of the structures. But this is only one interesting application. In the future, I believe, we will be able to use it for flexible accelerometers for soft robotics, because it's easy. You just smear it on the material, and it's self-assembled, and it's flexible and strong. Another interesting feature of cellulose nanocrystals is the fact that this is a highly thixotrophic material. What is thixotrophic material? For those that are not familiar, this is a phenomenon that if you have a material that has very high viscosity, like ketchup, for example, and you try now to put it on a burger, and if the cane is made of glass, then you cannot squeeze it, then it goes very slowly. So what are we doing? We shake it. When we shake it, we actually introduce shear force, and the viscosity drops dramatically. So that's the nice thing about thixotrophic materials. But once we apply it on a sandwich, for example, something really nice happened. It doesn't drop. It stays there. Why? Because the shear force is not there anymore, so the viscosity goes up very, very quickly. So that's the beauty of cellulose nanocrystal. It's a very, very thixotrophic material. So that enables us to take only 3% or 90%, 97% water, 3% cellulose nanocrystals, and put it in a syringe. And we actually took an STL file of butterscotch candy, and we printed from water at room temperature a 3D structure. The reason why we've been able to do it is because when we apply pressure on the plunger, despite the fact that the initial viscosity is very high, because of the shear force in the needle, the viscosity was dropped dramatically. So tiny droplets were now going out. But once they hit the stage, it solidifies. So that's beautiful, except this is like Jell-O, okay? Don't touch it. But once you lyophilize it, you freeze it and lyophilize it, it actually keeps the structure, which is beautiful. So now, taking all that together, about 7 years ago, we showcased in the Biennale of Architecture in Venezia, in the Israeli pavilion, a really beautiful structure that is theoretically, okay, this is a video, it's not real, a shelter in the desert that is mainly made of cellulose nanocrystals, but also the other interesting features that this material brings. So like superhydrophobicity, they enable us to collect the water dew in the morning, but then also make photovoltaic cells, which are partially transparent to collect some energy from the sun, and then use this energy, if you'll see in a few minutes or a few seconds, how we can use this energy to cool the water, and take this water and run it at the floor, so it will be nice for everybody to walk there in a nice environment. And of course, the beauty of this material, or all these materials, and when we don't want it anymore, we want to get rid of it, we just plow it into the ground, and because it's all bio-based materials. So this is still a dream, but we definitely realize the technology, and this is mainly a work of Dr. Michael Yani and Doron Kam, who just graduated, together with Professor Shlomo Magdassi. We've been able to develop a very nice material, we call it Daika wood, which actually use cellulose nanocrystals as the combining or the gluing material of sawdust that is now collected by the municipality every Wednesday in Israel, crushed, and instead of going to the ground for land filling, we can actually make a really beautiful material and products from that. And it's very strong. You can see here, we published it some time ago in Advanced Material, that they basically perform similar to the strength of oak. So it's quite a strong material. So I believe that using these materials, we'll be able in the future to avoid scenes as we see all these days. Now, I don't want to you to leave this room feeling that only plants are good. In fact, every kingdom in nature gave us really good things, including the insects. And I particularly like this creature, which I spent about 12 years of my life with it. It's called the cat flea. Now, why I'm so excited about cat fleas? Because they are world champions in jumping. They can jump as high as 100 times their height, which is remarkable if you think about it. The reason why it is remarkable, because it's the equivalent of a person standing in the middle of Manhattan and in a single jump go to the top of the Empire State Building, which is amazing, you have to admit it. So many people would like to have this ability. The question is, how can you do it? Or how cat fleas do it. So it turns out that cat fleas developed this beautiful protein that is called resilin. Resilin, in simple words, is the best rubber on earth. You can stretch it, you can squish it, and it holds the energy for a very, very long time. It doesn't lose any energy or almost any energy to the environment by heat or friction. And once you release it, boom, it brings back everything with very high acceleration. So that's how cat fleas actually make their jumping pad. They store this energy, and when they need, they release it, and boom, they jump on the top of the cat and do what they need to do. So, okay, resilin is great. Cat flea is a good source, so let's collect cat fleas. Bad idea, because they're jumpy, very difficult to collect. But that's the beauty of technology. All we need to do is just catch one, clone the gene, get it out, and express it, you're right, in tobacco. Well, we actually did it also in E. coli, also collaboration with a good friend of mine, neighbors, David Kaplan from Tufts. And we've been able to do some really nice and cool stuff with that, but I didn't tell you the entire story. In fact, in nature, it's not just the resilin. Actually, it's a nanocomposite that is made of nanochitin fibers that the resin attached to these nanofibers using a chitin binding domain. So you have the elastic region, and then you have the chitin binding domain. But we prefer cellulose for all the obvious reasons. So we simply replace the chitin binding domain with a cellulose binding domain. So now we have the opportunity to make composites with cellulose nanocrystals. So the guys in the lab said, hey, you know, we have cellulose nanocrystals, which are really tough, really nice material. We can make foams. What will happen if we will take this foam and add a little bit of CBD resin? So you can see a short video here. On the right-hand side, it's cellulose nanocrystal foams in water. I squish it with a spatula, and you can see it has only plastic deformation. So not so exciting. On the left-hand side, it's actually the same thing, except that we have now some resin. And sure enough, you can see a very, very different mechanical properties. So you can do it again and again and again. By the way, every time when I show that, at that point, people smile. And I think that it has to do with our childhood. You know, the Humpty Dumpty. In Israel, we have something called Nahum Takum. It's about the same. And by the way, I found only one place in the world that they don't smile. Because I had the chance to talk about it in different places in the world. But I'm not going to tell you, because I know this is going to be a business opportunity to sell them Humpty Dumpty. Anyway, the mechanical properties are really amazing, and you can see it. But interestingly enough about the resilin is that, in fact, it's a photopolymer. In nature, actually, the material is being cross-linked using an enzyme, which actually form the dityrosine bridges. But we actually were able to take this protein, just adding a photoinitiator and using a nanoscribe, which is a 2-photon printer that enables us to print submicron structures. And by doing that, Doron was able to print a resilin made, insect. and you can see here it's a sub-micron resolution. So what can you do with this resilin? A lot of things. We're now working with On. It's a Swiss company that interested in making sports shoes with some better performance for athletes. But you remember the collagen I told you before? We asked ourselves, what's going to happen if we'll take a collagen fiber and we'll add a little bit of resilin, only 5% resilin? What's going to be the mechanical properties? And you can see clearly in the instrument test, in tensile stress, that we've been able to increase the toughness by 380% and 300% increase in strain at break, which oddly enough indicates that in the future, once we have artificial tendons or ligaments from that material, the patient will have better performance after the surgery than he had before the injury. But this is just one of the things we could do. I think you can use your imagination to do a lot of interesting things with it. So what's for the future? I believe that in the future we will be able to produce almost anything in plants. So not just collagen or resilin or many other things, nanocellulose made by itself. But in fact, everything that nature made us and we have the sequence for production, we can probably produce in plants. And then we will purify these materials and use modern technologies like 3D printing to print almost anything, even a heart. Now, this heart is not going to be as good as we can get from a donor. It will be better because it will be made from brand new materials, all right? Well, admittedly, this is probably not going to be in my lifetime, but I believe that in the future we will be able to do that. So I'd like to wrap up my talk by quoting another smart guy, and his name was Pavlov. And Pavlov said that if you want a new idea, you should open an old book. And I say that the book was written. It was written over billions of years of evolution. And the text is the DNA of life. We don't have to reinvent the wheel. All we need to do is read the text and start our progress from there. Thank you.
Any question? Please, Chris.
From the perspective of being a lab tool, I understand why tobacco is a very straightforward host to genetically manipulate. If you were thinking, kind of, if you had the ability to modify any plant with that type of capacity, does tobacco still stay the optimal protein producer, or do you think for some of these applications it would be better to go to something like soybean or some other plant?
Thank you, Chris. That's a great question. In fact, you have to put things in perspective of time. 18 years ago, the idea to clone 5 human genes in a single plant and optimize the expression of the 5 was crazy enough. And therefore, that's why we picked tobacco at that time. Today, we do other things, like, for example, we produce dairy proteins in oil seed plants. Our target, by the way, is, we actually work with model plants like Arabidopsis and Camelina, but our partners in Cyro in Australia actually helped us to put it in safflower. Safflower is quite recalcitrant for transformation, so it's not easy to do. But definitely, if you want to produce something at large scale, a commodity protein, I would say that tobacco is not the organism. For coal plant, collagen is so expensive. Humanized antibodies are so expensive. That's not a problem. But I completely agree with you.
So in clinical use, they use UV light to cross-link the collagen in eyes, the corneas. So, and I noticed that you used polyvinyl alcohol, I think. So have you tried, is there a difference between cross-linking using the alcohol versus UV light?
Okay, so first of all, you're right. Cross-linking of cornea is done with UV light and riboflavin. They actually put some riboflavin as a photoinitiator, but it's a very, very slow process. It actually If you ever experienced that or saw it, you're sitting there and they shine UV light into your eye. It's not the best thing. We have the option to make it much faster, the cross-linking, which is very important when you do 3D printing. It's particularly at high resolution. So what we actually did was we metacrylated the collagen. And when you metacrylate it and now combine it with a photoinitiator, like IrgaCure or LAP, whatever you want, then the cross-linking is instantaneous. So it's much better. But you're right. Collagen can be cross-linked with a simple riboflavin, but it's going to be slow.
Oh, thank you. It's a very exciting talk. Feels like really making impact in real life and bring nanotechnology. So I guess my questions are, the first one is, you're using cellulose nanocrystals rather than cellulose nanofibers, but cellulose nanofibers might be even stronger in terms of strengthening the cotton or for being the coating themselves. So could you tell me why you're choosing the nanocrystal rather than the nanofibers for that? And the second question would be I know that cellulose nanocrystals and biomaterials are great for their water solubility or water dispersibility, but it can also be a problem. For example, you can create beauty in a desert because it barely rains, but in more other circumstances it might be less ideal. So would there be any ways around to make them more waterproof with their build?
Okay, so basically two different questions. So first of all, no offense to cellulose and nanofibers. They're useful for a lot of things. You're right. If you want to use them, for composites, the fact that they are longer, the fact that they contain also amorphous regions is actually a good thing. But if you want a chiral nematic structure, you can't do it with cellulose nanofibers. You must have cellulose nanocrystals. And particularly if you want to make either structural colors or you want a good charge separation, if you want piezoelectric properties, cellulose nanocrystals is by far better. As for the water solubility of it's not soluble, it's actually suspended in water. Yes, but I've showed you with this very simple chemistry, you can actually make it quite hydrophobic. Acetylation, whatever. So there are simple solutions for that.
Yeah, thanks for the very interesting talk. I have a question about the wound healing with collagen. You showed that in a matter of 3 months, a very large wound closed completely, which is amazing in and of itself. But I was curious whether you see tissue regeneration or scar formation and why.
Okay, so great question. It's actually, particularly for old people, it is definitely scar formation. There is no tissue regeneration. But I can share with you some information. We didn't really publish it, but because this was done off-label by a doctor that had a young person that was bitten by a dog, and for about 1 month there was no recovery. And so he debribed it, and doctors can do off-label. We cannot publish it, but he did it. And the results were remarkable. And there was absolutely no scar after 4 months or so. The reason for that is because he was young. And because he was young, he still had a lot of mesenchymal stem cells. When we get older, we don't have enough of those. But I believe that in the future, once cell therapy will be more popular, people will use combination of both stem cells and the collagen together. So it is possible.
Any other questions?
You showed that cellulose nanocrystal-based polymers have very strong material properties. did you ever test what happens if they are discarded into the environment, and if so, what half-life do you expect?
So what I didn't mention to you is that it actually, because it's a polyvinyl alcohol, at the end of the day, it is water soluble, which is good for the environment. It's not good for the sailor. Okay, so it's a very, very strong material, provided that there is low humidity. So I think so that's a real issue. We're thinking of make some coatings that may repel the water. Actually, just before this meeting, I had a meeting with, where are you? I saw you there. One of your, oh, yeah. Maybe we can do something about it, right? So I think there are some opportunities, but yeah, it is water soluble.
I really like the quote you had at the beginning about the Stone Age not ending when we ran out of stone. And I think it's true, but we also currently still use a lot of stone. So as you're, right, and we've come up with sort of new materials and new applications for them. So I'm curious, as you look to the future, do you see a lot of these materials sort of replacing metals or plastics in existing applications, or more as sort of enabling new applications with things that those materials can't do?
So I think both. I think both. I'm not here to say that all the good materials that we developed so far with organic chemistry, polymer science, are not going to be used. But what I'm trying to advocate is really two things. First of all, that natural material gives new opportunities for new materials and still being sustainable. And the really amazing materials that we produced by polymer chemistry, I hope that we will use them just for what they're needed, because they're, from the environment point of view, they're not really sustainable. That's my point.
Okay, so I think with this we can conclude. Thank you very much.